and I'm going to talk briefly about the importance of marine and freshwater spawning for coastal pike in Western Estonia. As a lot of previous presenters mentioned before me, broadly speaking, we can say that pike has three different life history types. Firstly, there is an anadromous type where fish spawn in freshwater, but most of the feeding and growth is done at the sea. Then there is a brackish water resident type where all life stages are spent in brackish water. And thirdly, there is freshwater resident. If we talk strictly about coastal waters, then the freshwater resident is more or less out of the picture. So what do we know about the situation in Baltic Sea? What is the pike origin story? One study from Sweden found using otolith microchemistry that 45% is of freshwater origin. On the other hand, a more recent study from Germany in Rügen Island found that only 6% is of freshwater origin. This means representing anadromous life form. But what is the situation in Estonia? Here is a map from 1970. It shows semi-speculative pike spawning grounds in Novemeber Sea area. According to this map, pike spawning grounds are quite extensively distributed in Novemeber Sea. But this source doesn't give exact information how those areas have been identified. Probably pike with running gametes were caught from those areas. So it is unclear if those fish were really spawning on those sites or really they were just on their way to their real freshwater spawning grounds. From more recent time, there is a study using otolith microchemistry, but with rather small sample size from Matsalu Bay, showed that 5 fish from a total of 28 fish were hatched in brackish water. And from all that, we got an idea of an even larger study. So for this larger study, we gathered fish from broader coastal waters of Western Estonia. A total of 498 adult pikes otoliths were used for microchemical analysis. Samples were obtained mostly from commercial fishery, but also from coastal monitoring service. And fish were caught mostly outside of the spawning season. So in the lab, the first step was, of course, otolith removal. Then the otoliths were ground to the core and polished. We used the strontium-calcium ratio and strontium isotopic ratio as a markers for hatching biome identification. For next slide, I'll make a brief digression to the theoretical background. Firstly, strontium calcium is high in seawater and low in freshwater, at least in Estonia and in adjacent areas. Strontium isotopic ratio, on the other hand, is characterized by the universal global seawater value. And in Estonia, freshwater values are higher than that. So here you can see an example of anadromous pike otolith microchemical profile, where blue line represents strontium calcium ratio and red line isotopic ratio. On the far left, representing the otolith core, is so-called maternal influence zone. And this zone in this zone, the otolith material is mostly derived from the yolk. In this particular case, we see that there are high strontium values and low isotopic values, indicating that this fish had a mother from a brackish habitat. Right next to the core is the area which represents the hatching and early life biome. From that, we can obtain natal signature. In this case, we see that this pike has hatched in freshwater, indicated by low strontium values and high isotopic values. Natal signature itself is a part of larger freshwater rearing phase, which is ended in this case when pike enters the seawater, which is usually characterized by sharp increase in strontium values and decrease in isotopic ratios. In this current study, we're interested in natal signature, to be exact, if the natural signature is from freshwater or from brackish water. So let's see our results. In the western Saaremaa coastal sea, right next to the Baltic proper, majority of the pike were of freshwater origin. Only 3%, okay, actually only one pike was hatched in brackish water. From the Saaremaa southern coast, we had larger sample size. And in this area, again, most of the fish were of freshwater origin, but share of the brackish water origin was greater than in the west coast. Moving to the Novemeber Sea area, where we sampled majority of the fish, the general picture is more or less the same as in Saaremaa southern coast, where freshwater origin was in clear majority. Now, let's focus our attention to the Novemeber Sea area. Are there any differences in smaller spatial scale? If the sample is broken into small subsamples, then we can see that there are some sites where brackish water origin has greater share, or even in one place is dominant hatching biome. On the other hand, there are a lot of green balls on the map showing a lot of freshwater origin. But how are our results accordance with the spawning grounds map from the 60s, which I showed earlier? I would say that there is a partial agreement. We can see that in this southern Hiiumaa coast, our results match with the map, but not much elsewhere. Interestingly, this time in Matsalu Bay, we didn't identify not a single fish that was born in brackish water, although previous study found that 18% of the analyzed fish were brackish water recruits. And as mentioned previously, most of the pikes were sampled outside of the spawning season, but in some areas denoted by the asterisks in this map, there was some sampling done also during the spawning season. If we remove those fish sampled during the spawning season, we get a picture with a little bit less green. If only pike sampled outside the spawning season are included, then we get nice round numbers with a share of brackish water recruits somewhat increases, so that 1/5 of the fish are of brackish water origin in Novemeber Sea. But what about pike sampled during the spawning season? Here we see that there is no big surprises yet, as most of the fish were sampled near well-known freshwater spawning sites. Almost all fish were of freshwater origin. So far I have talked about the situation in the sea, but what is going on in the adjacent freshwater spawning grounds? Here I put those dots as a background info from previous slides, and those donut-shaped circles show prevalence of anadromous and freshwater resident progeny in different freshwater water bodies adjacent to coastal sea. Now here is important to note that now we are speaking of maternal origin, not natal origin, as all those fish were born in freshwater. As you can see, there are lots of blue on those circles, showing that majority of those sites are dominated by anadromous spawners. This furthermore emphasizes the importance of freshwater spawning for coastal pike in Estonia nowadays. Okay, let's finish with the Novemeber, and let's have a quick closer look at the southern coast of Saaremaa. Here all the fish are sampled outside of the spawning season, and we can see that there are striking differences between neighboring sites, as most of the bays have strong freshwater origin dominance. There is one bay where all the pike were of brackish water origin. This shows that there are areas where brackish spawning is dominant, and it can occur in relatively small spatial scale. To conclude, our study demonstrated that freshwater recruitment has high importance for pike in Estonian coastal waters, and that most of the pike in the sea represent anadromous life history type. This leads to the notion that well-functioning freshwater spawning grounds are crucial for successful recruitment, and those sites should be protected and well managed. Brackish water recruitment is in minority in most sea areas, but those areas with substantial brackish spawning still occurring should be under protection and managed accordingly. There may be a couple of reasons why the share of brackish water spawning is low as it is right now. The sea spawning pike may have been overfished in past and possibly also right now. Furthermore, there may be problems that can be associated with eutrophication. Also, sticklebacks may be involved, or combination of those factors is the real cause. On the other hand, we don't exactly know if brackish water spawning was historically any different from what it is right now. And lastly, there can be sharp differences in small spatial scale regarding pike hatching biome, indicating that brackish spawning could be quite restricted in space. And I think this is it with my brief overview of this matter. And thank you for your attention. Now it's lots of time for questions.
Thank you.